I'll say a few words for the how to pick me, because that's how it works, this thing. Thank you very, very much for everyone who came and logged in online, and to our great host from the Bio Foundation who invited me. A lot was said, just to make it clear on the postdoc at the Max Planck Institute, nothing more. And today, I would like to tell you what I think is an interesting story about how our understanding of immune systems and what immune systems look like and how they behave in genomes and how this behavior can help us find new parts of immunity has led me to the current project that I'm working on. So the thing doesn't work, so thank you, Thomas. No? Yes. Oh, it's going to be very hard to do this. Yes. Give me a second. Let's see. That works? Yeah, now it works. Great. So, all organisms, it's fine. All organisms, as we personally know from our own experience, face the threat of infection by pathogens. This is true for every single living thing on this earth, because there's always another living thing that wants to eat you. So we have a personal experience with this when we were sick with viral diseases. Usually when I give this kind of talk to a broader audience, I need to explain that also plants can get sick, but since we are on one hand, I don't need to specifically iterate it because everyone knows it. But another aspect of infection that many people kind of forget about is that the most abundant organisms on Earth, which are bacteria, they are the most ancient, the most numerous, and also the most diverse, they also get sick. And the things that make them sick are bacteriophages. Bacteriophages are viruses that infect bacteria as hosts. They are kind of evolutionarily related to the rest of the viruses that also infect us in many ways. But because bacteria are the most abundant organisms on Earth, their viruses are the most abundant viruses on Earth. And it's often said that they are the most abundant biological entities on Earth because it's hard to say that they are organisms because they are viruses. But anyway, they cause a huge evolutionary pressure on bacteria to deal with them. And this evolutionary pressure, this most ancient war between bacterial viruses and bacteria, has led to the development of bacterial immune systems. And two immune systems of bacteria are, for example, restriction enzymes and CRISPR-Cas. Now, I guess no one in the room is specifically a bacterial immunologist, but I believe that anyone that does anything with molecular biology have heard about restriction enzymes and CRISPR-Cas. And the reason is that these molecular mechanisms that were invented by bacterial evolution to deal with viral infection, after the discovery and understanding, have been adapted as tools to do this amazing thing, which is to cut and edit DNA and genomes. So the fact that the natural evolution of bacterial immunity has given rise to such amazingly powerful tools brought up the question, how can we find additional such immune systems that might later be evolved into additional super powerful molecular tools? And that was the topic of my master's and PhD studies at the Weizmann Institute of Science in Israel. For this very long period. So let's dive into how did we try to answer this question. And the way we did it is by using a very interesting observation that at first we didn't know how to explain, and to tell the truth, I still do not know exactly why this is the case, but it is the case. These kind of representations of genomes is something that I will show repeatedly in this talk, so let's just make it clear. The line is a piece of a genome, so a piece of consecutive DNA of a genome of something, and these smaller arrows represent the genes encoded on the genome. It's the common representation of the genetic loci. And the way it works with bacterial defense systems is that it was observed empirically that if you find a defense system encoded at a given position on the bacterial genome, there is a good chance, statistically significant, that in the vicinity of that defense system, there will be another defense system. And next to it, another defense system. So it seems that the way bacterial genome evolution works is that they clump these systems together physically on the genome. So they just are encoded one next to another on the bacterial genome. And this simple observation gave rise to the idea that if we find genes that are repeatedly in different independent genomes, repeatedly co-localized with known defense systems, maybe they are also defense systems. And it turned out that this is indeed the case. So this is a very simple concept, but it proven to be extremely, extremely powerful. Because if we look at what we knew of classical bacterial immunology that contain restriction enzymes and CRISPR and maybe a few toxin antitoxin systems and a few more anecdotes, using this systematic screening based on genomic association with known defense systems, the world of bacterial immunity really exploded in the last since 2018. So it really revolutionized the field, the fact that we could predict bacterial immune systems based on their genomic property. One thing, so okay, this is a zoom of things, and each one of them made for a very, very nice paper, usually very well accepted, and propelled the careers of many people, including myself. And what I would like now to focus on that we learned from this huge explosion is one particular aspect of it that I spent a few years studying. And this is just one of these defense systems, which is here. So this system was named Thoeris. We named it Thoeris. We named all the immune systems that we found after ancient goddesses and deities of different mythologies. So Thoeris is the ancient Egyptian god and goddess of fertility. While working on it, I had both of my daughters born, so I guess it works. It's not an especially common immune system, so it's found in about only 4% of the bacterial genomes. But when I saw it, when we found out these genes in defense islands and proven that there are immune systems, I immediately want to work on it. And the reason is that it contains two genes, which is good because only two, you can work with it. And the second gene contains a tier domain. And tier domains, for anyone who took Immunology 101 or anything that has to do with immunology, we recognize that tier domains are canonical components of innate immune systems of animals and plants. But their existence in a bacterial immune system came as a big surprise when we first saw it. Because generally speaking, we don't think of immunity as being shared between bacteria and eukaryotes, right? We have antibodies, but there are restriction enzymes, there's nothing to do with it. And when we first observed that bacterial immune systems contain tier domains, I was intrigued. So we started working on it in depth and trying to understand what is going on. And we were lucky enough that we did it around 2018, 2019, because 2019, there was an amazing set, 2018, 2019, there was an amazing set of discoveries on how eukaryotic tier-containing proteins are working, and especially in plants. So in plants, tier domains are part of NLR, receptors, immune receptors of plants. And it was shown by one and Postfield in a back-to-back amazing set of papers that once these receptors detect the invading pathogen to the plant, what they do is that they multimerize, activate the enzymatic activity of the tier domains, which were not known to be enzymatically active at all before, because most tier domains in humans, for example, are not enzymatic. But in plants, they are enzymatic. They become enzymatically active. They generate a signaling molecule that cause let's say, programmed cell death of the infected tissue, and thus contain the infection of the plant. This discovery made us think, is it possible that also the bacterial T domains also produce secondary messenger molecules that would cause this kind of programmed cell death and contain the infection of the bacteria? And it turned out that this is exactly the case. So long story short, we found that the Toeris proteins, the T proteins of Toeris, detect the infection by the virus or in the bacteria, generate a secondary messenger molecule that is binding to the second protein of Toeris, which then causes the death of the infected cell. So conceptually, and both conceptually and biochemically, these systems work really in parallel. So this was published in 21 and really made me want to keep working on this aspect. So this discovery basically made us understand that T-domain proteins that have been studied as part of eukaryotic immunity for decades actually exist throughout the tree of life. So the discovery was in Drosophila, actually in development and not in immunity, but now we know they are also involved in immunity in Drosophila. Funny story is that they were discovered in Drosophila in Tübingen, right where I currently do my postdoc. They were then shown to be involved in immunity in plants. Actually, the first case of T proteins being involved in immunity was shown in plants and not in animals. Later, they were shown to be part of toll-like receptors, which are the canonical innate immune receptors of humans. And our discovery added that already in bacteria, they are functioning as immune components of systems. And this was, I would say, a really interesting conceptual breakthrough. The fact that you can think of components of immunity from eukaryotes as functioning in a very similar manner also in prokaryotes. But T proteins are not alone in this thing. So after basically at the same time where we discovered this connection of field, there was a set of discoveries in Bottom Sorex labs where I was working and in other labs that showed that the concept of conservation of immune components between bacteria and eukaryotes is actually a general rule. So not to go into the molecular details, but those of you who know of the SIGA-STING pathway in humans, we demonstrated that it exists in a pretty similar manner also in prokaryotes. There's a protein that's in humans called SAMHD1. Anyone who works on AIDS actually probably knows it. What it does, it depletes the nucleotide pool from the cell to prevent viral genome synthesis. And it has homologs in bacteria that do pretty much the same thing, deplete the nucleotide pool to prevent viral replication. And there are proteins called viperins that generate and chain terminating nucleotides that prevent the synthesis of RNA viruses, basically kind of like we do Simon sequencing when you put a nucleotide that cannot be elongated. So the same story happens both in humans and in bacteria. And the very basic NLR receptors that are the mainstay of plant immunity and also of inflammation in humans actually have bacterial counterparts that function in a similar way. So it seems that it's a general trend that many components of immunity in eukaryotes were actually inherited from the last common ancestor of eukaryotes, who was a bacterium, or an archaeon, to be precise. What we also noticed after uncovering this amazing diversity of the immune system is that the set of enzymatic activities and the set of domains that we see in immune systems across the tree of life, both in prokaryotes and in eukaryotes, it's a defined set. You don't find everything. There are things, there are a lot of biochemical activities and protein domains that you find repeatedly in multiple immune systems, both in prokaryotes and in eukaryotes. So Alvin wrote it beautifully that you can look at the different immune components as targeting the different parts of the central dogma of biology. So you have a lot of nucleases, RNases, and DNases that would attack the genomes of the pathogens. You would have a lot of nucleases that would cleave nucleotide metabolites that are essential for life anywhere, and therefore could be used also as immune protein. On the same line, you can have peptidases, but you can see the general trend. These are not like any, for example, most of the domains that catalyze normal metabolic reactions are absent from immune systems. But on the other hand, you see again and again and again hydrolases that cut different essential components of living things. And this is because immunity, in essence, is detecting the enemy, differentiating the enemy from yourself, and then destroying the enemy. And since the enemy is a living entity, there are many, but limited, set of things that you can do to it to kill it, which is basically to cleave its genetic material, to cleave its protein, to cleave its lipids, or to destroy its cellular components. So, based on both the conservation of immunity between prokaryotes and eukaryotes, and the understanding that the grammar of the immune system is something that we can learn and understand and predict, there were a set of, I think, very beautiful papers that applied it to use what we know from prokaryotes in eukaryotes and from prokaryotes in eukaryotes. So one that I would like to highlight is a very recent paper by Vandenberg. They did something that is beautifully simple yet complicated. They took all known immune systems from vertebrates and a bit of plants, collected all of them together, and basically searched for homologs in Pseudomonas, which is a bacterium. Pseudomonas is an excellent bacterium to work with because we have huge collections of it. We have thousands of fully sequenced Pseudomonas genome because it's a clinical relevant pathogen. And they basically just searched for homologs of eukaryotic immune systems in the multiple Pseudomonas genomes that we have. The things that they found, again, are not randomly functional. They have clear enrichment for things that we know are involved in immunity. So it's true that it's biased to their starting set, but it really captures the fact that immune systems have their own grammar of domains. So you see a lot of things that deal with DNA and RNA, and then a lot of hydrolases of all sorts. But when they took many of the homologs that they found in Pseudomonas and tested, do they have antiviral activity in Pseudomonas, they were able to identify multiple new Pseudomonas immune systems that bear some resemblance to eukaryotic immune systems that we didn't know existed in both domains. So this really is an amazing example of how you can take what you know from eukaryotes and find it in prokaryotes. But this could also be done the other way around, when in this study by Korean, a very, very new paper that was published just last week, where they took a set of all known antiphase systems of prokaryotes, searched for it in eukaryotes, and when they found the homologs, they were able to demonstrate 3 human components of immunity that were unknown to be involved in human immunity. They chose humans because it's a simple model. Human cell line is a simple model for eukaryotic viral infection, and this again demonstrates the power of, once you understand the grammar of immunity, and once you understand the evolutionary connections between things, it allows you to find discoveries on both ends. So, to sum up the first part, there are 3 things that I and the field have learned from these studies in the last, let's say, 5 years, 6 years older. One is that immune systems have genomic signatures. They can be identified using, in prokaryotes, the defense island hypothesis. And this if you can do it, it's extremely powerful. If you find the genomic signature, then you can really look at genomes and find new immune systems. The second thing is that they have their own grammar of domains. They have a set of domains that they use, and this can really be a powerful tool. If you find genes of unknown function that you ask yourself, is it immune or not? The presence of suspected domains in it is a very powerful predictive tool. And the third thing is it's not that they just use a defined set of domains, but these domains serve in immune systems from the dawn of life, from when life were only prokaryotic and were inherited by the early eukaryotes into their own immune systems. And at this point, I did a weird career change, I would say, or career change of direction. And that was to transition from working on bacteria, which are very small, grow very fast, and very easily edited, to plants. I have absolutely no good career explanation to that, except for the fact that I really like plants and I wanted to work on them. But from any practical aspect, it's a bad idea. So do it only if you really love plants. And the reason I did it is it's really just because I love plants. The beauty of plants and the diversity of plants is what got me into biology for the first place. I was a botanist. I thought, okay, botany is biology. I want to study biology. Found myself in biochemistry, which is also beautiful in its own way. But plants are really fascinating for me, and I think for many people who work in this amazing campus that we are in. They do have several differences from bacteria. First, they have much larger genomes, which is a big problem if you want to do genomics. They are big, so it's hard to fit 10 to the 11 of them in a test tube. And they grow slower than bacteria, so you can't think of an experiment today and run it tomorrow, but you need to wait for the Arabidopsis to make seeds in 2 months. But on the other hand, they have amazing metabolism and can do things that heterotrophic organisms like ourselves could not even dream of. They provide us food, which is good if you like to eat. And they are the basis of all ecosystems on Earth, almost, because the photosynthesis of plants is what's driving the natural world. So they are both important for practical reasons, but also if your motivation is to understand the natural world, which is more my motivation as a basic scientist, they are critical for that. So I did this transition, and I was hoping to combine what we learned on immunity from the prokaryotic world into the plant world, and not into the animal world, because in the animal world, many other people do that, and I'm also less interested. So one thing that we learned that I want to utilize is the fact that immune systems are conserved, as I just described before. So can we find new homologs of bacterial immune systems in plant genomes and not in animal genomes? Our knowledge of plant immunity is much limited compared to our knowledge of animal immunity, because we are animals and we study it ourselves. But there is a lot of plants have amazing immune systems, and I thought this new direction could lead to interesting observation. And the other direction is actually the complete opposite. So I was talking a lot about how immune systems can be conserved across the tree of life, but actually immune systems are also the most diverse part of any organism. So can we use this diversity to identify a genomic signature conceptually parallel to the defense islands of prokaryotes that would allow us to genomically predict immune systems in plant genomes? And this interplay is what is keeping my research interesting for me. So without getting into the specific details, one of the things that we did is that we were able to take an immune protein, to find an immune protein of bacteria that was only recently discovered in this huge expansion of immune systems that happened in the last few years, and find a plant protein that we really believe is homologous to it, and therefore may be conserved. And now the question is, if we find this amazing homology, can we also demonstrate that it is also involved in immunity in plants? And for that, we are doing the actual experiments in the lab, mostly in the model system of Nicotiana betamiana, that is very easily infected by viruses. And we try to test if this conserved protein is also part of the plant immune system. We have encouraging results, but not final enough to fully share. But this direction has been surprisingly successful in this in the search for new components of the plant immune system. But what I want to focus more is during this talk is on the fact that the genomic signature that we used in bacteria was actually this was the silver bullet of the entire thing. The fact that we realized that we can predict immune systems based on their genomic localization, the genomic feature was the thing that drove the whole thing forward. And I'm still very much interested in asking, can we find a parallel thing in plants? We know that in plants, it doesn't work like in bacteria. Plant genomes are much more messy than bacterial genomes, but maybe we can find something else. So how can we identify genomic signatures of plant immune genes? And I think the key to that is to understand that in general, immunity-related loci, genomic loci, show a very strong diversity signature. What do I mean by that? If I take the genome of myself and any other person in this room, and we compare it, it will be 99 point several digits identical, because humans are very homogeneous species. But the one thing that will definitely be different between you and me and any two human individuals is the HLA locus. The HLA locus encodes for immune receptors, immune presenting receptors on cells. And this is the most diverse part of the human genome. It's more diverse than any other group of genes, maybe except transposable elements. But this is not specific to humans. It is also true for bacteria. If you take any two E. coli accessions, and we have currently thousands of sequenced E. coli genomes, and you compare them, you can very easily find two E. coli genomes that are completely identical, has very, very few SNPs between them, but contain different immune systems in specific loci. So the fact that immune systems are the most diversified part of the genome is a well-established observation. And the reason for that is that immunity is a race between two things that both run. So if a plant or an organism adapts to high temperature does not try to adapt to this organism. But if an organism adapts to overcome a pathogen, the pathogen definitely tries to adapt to overcome the immunity. And this creates what is often termed as a red queen arms race, based on the quote from the Red Queen, that means that it takes all the running you can do to keep in one place. Since both sides are running, if the host is not changing in a way that matches the evolution of the pathogen will win. So immune genes are under this constant pressure to diversify. And if we take it now to plants, so the main thing we know on plant immunity is the involvement of plant family of immune receptors called NLRs, nucleotide binding leucine rich receptors. And these NLRs are known to bind pathogen components directly. And you can look at it and understand that if both sides are trying to play here, then once there is an immune receptor in orange that binds a pathogen effector, the pathogen is under amazing evolutionary pressure to change the effector, and then the host, the plant, will need to come up with a new NLR to identify the new variant in the effector, and so on and so forth, ad absurdum. And the way this arms race manifests in genomes is that if you look at the collection of, in this case, Arabidopsis genomes and compare them to one another, so as before, each line is a piece of the genome, and the genes are marked by these rectangles. You can see that for the most part, the genes are the same because these are Arabidopsis plants. These are all Arabidopsis plants, so they look the same, they do the same metabolism, they're very closely related evolutionary, same species. But even within the same species, once you get to a locus that contains NLR genes, it gets messy. So each accession, each Arabidopsis that was collected from a different place in the world will have almost identical genome to all the rest, except for the loci that encodes for NLR genomes. And this hyperdiversity in specific loci, I believe, could guide the discovery of new things. And with that idea, I joined the Department of Data Weiger at the Planck Tübingen because Detlef was a pioneer of looking at the natural diversity of Arabidopsis and was one of the driving forces behind the 1001 genomes collection that includes way more than 1001 now, Arabidopsis accessions that were collected throughout the world, mainly in Europe, because it's a mainly European species, but also in Africa and in Central Asia and in the Far East. And among these thousands of accessions that were collected, at least 300 have fully assembled high-quality genomes, which gives an amazing resource for looking at genomic diversity, a resource that is very hard to get when it comes to plants outside of Arabidopsis. So if we look at the genomes of these accessions, as was beautifully shown in a paper from the lab that was published by Tizem Moore et al., also last week, I think, or 2 weeks ago, highly recommended. Again, if you look at these three accessions, you can see that in this part, they pretty much look the same and code the same genes. Then starts the NLR region, which is messy, and then starts a normal region of the genome that again look the same. And if we look at a larger collection, you can really see that if you can define these regions based on the edges of the less conserved genes on both sides, you could really see which regions of the genome contain these hyperdiversified genes. And this is not that different conceptually from the defense islands that I was talking about before, except that in plants, we know the NLRs are immune in orange, and we have absolutely no idea what the blue genes are for the most part. And this is one of the things that I'm trying to understand. So can we use identifying these hyperdiversified regions in plants to find new genes that are involved in immunity, because very few evolutionary pressures outside of immunity would drive this kind of super rapid diversification. And the other direction that can be taken is to ask, okay, so we know NLR regions are hypervariable, but can we find other hypervariable regions that are not NLR containing? And the answer is definitely yes. We do find such things that contain very surprising genes that we had no idea that might be involved in immunity. And this is another set of projects that I'm working on with my students to try to find places in the genome that look like this and ask, what is this? What kind of genes undergo this kind of dynamics? I want to just briefly reiterate the challenges here. And the main challenges here is that plant genomics is orders of magnitude more complex than bacterial genomics. Plant genomes are much larger, so we're talking about several orders of magnitude larger. Even if you work in Arabidopsis, which is one of the smallest plant genomes, it's already in the order of hundreds of megabases, while the normal bacterium is 5 megabases. So this makes things much harder. And not only they are larger, they are also repetitive. And if they are repetitive, it means that with short reads, you can hardly make sense of them because they contain repetitive regions and the short reads look the same and you don't know how to make it. So it's very hard to assemble them and then to annotate exactly the genes. This is not a problem that is unique to plants, but is true also for animals. But one could say that maybe it is worse in plants than in most animals. And the other point is, since these genomes are so hard to assemble to begin with, usually what you do is you assemble a reference genome, and then if you sequence another genome, you align what you sequence to the reference genome, which works perfectly well for 99% of the genes, but does not work at all when you're looking at genes that are by definition different in different genomes. If you try to align them to the original genome, you would just not see them. So, and to make things even worse, immune genes are not only reference accession specific, but are also repetitive among themselves, because this locus of NLRs is actually one NLR that was duplicated and diversified. So this gives you repetitiveness within non-reference region, which is basically the hardest thing to solve when you try to make a new gene. Luckily, these problems are now being solved. So we have the long-read technology and the new assemblers that are finally allow us to overcome these things. So now making a new plant genome from scratch for Arabidopsis, it's like not easy, but absolutely feasible. And that's how we got to the level that we have 300 really good ones with long read, fully polished, nanobio, whatever you need, that we really can see these repetitive regions fully assembled as they are. This was impossible up until very recently. And the second thing that is completely unrelated to plant science, but this is what I bring with me from the bacterial side, is that we start to understand the grammar of immunity and what immune systems look like and how they behave in genomes and what do they contain. And I believe that the combination of these two things put us in a very good position to dive into these new genomes and try to explore what lives in these islands that we couldn't see before we had the technology to see them. So that's the things that I'm working on and that's driving me. And with that, I would like to thank first my scientific mentors, Rotem, my PhD advisor, and Detlef, my postdoc advisor at the Max Planck Institute and the Weizmann Institute of Science in Israel. My amazing team of students who work with me, which is really unbelievable. So Ali, Lucas, Helena, Sandra, Nicholas, and Julian just left, but I am in general recruiting. So if someone finds this super interesting, then please get in touch. EMBO and HSP for my postdoctoral fellowships that support me, and especially my family who followed me across the sea, not ocean, but to Germany to be able to pursue this line of research. And thank you for listening, and I'll be more than happy to take questions.